Okay, so now what we did yesterday was we basically discussed RER, Golgi apparatus, lysosomes, smooth endoplasmic reticulum, and we finished our yesterday's lecture on mitochondria. So we discussed the process of protein synthesis a little bit more, and then we talked that once protein synthesis is complete in the RER, it goes to the Golgi body and then it undergoes a little bit of more modification. And we understood that entire idea of how a protein is modified by attachment of some other molecules to it. And then once we have that modified protein is then our final product and it is packaged into vesicles. And if it is required by the cell, those vesicles will be used within the cell, or else the Golgi would transport those vesicles to the cell membrane, and then the cell membrane would then transport those vesicles out of the cell. So it can go to whatever region it wants to, or wherever those proteins are required. We also understood that mitochondria is basically the powerhouse of the cell, and we talked about all the labelings and all the compartments of mitochondria and how it is a double membrane structure, and it has its own circular DNA, it has its own 70S ribosomes, and because it has its own genetic material, so that DNA can be converted into mRNA in the process of transcription, and that mRNA then can go on the 70S ribosomes, and then it will form its protein or the polypeptide chain by the process of translation. So we said because it has its own protein synthesis machinery, it has its own protein factory, so it is self-replicating organelle. Okay. That's about it. The major role of mitochondria is basically the production or the synthesis of ATP, which is our energy. This is the energy currency of our cell. So whatever processes in the cell or in the organism's body which require energy, that energy is basically derived from ATP. And where is this ATP coming from? This ATP is basically coming from the mitochondria. And thus, mitochondria is basically the organelle which is very important for the energy processing and the energy synthesis of our body. Okay, so that's where we concluded our yesterday's class in the chapter of mitochondria. Now let's come and discuss another very important organelle, which is chloroplast. Now, when we talk about chloroplast is present in plants. So chloroplast would only be seen in those cells which are autotrophic. Now, what do I mean by autotrophic? Autotrophic is basically a mode of nutrition. What do I mean by that? When we talked about autophagy and autolysis yesterday, we said auto means by its own self. खुद से कोई चीज खुद ब खुद हो जाना, वो that is where we derive the word auto from. So autotrophic are those organisms, they are those organisms which can make their own food. Like for example, when we talk about humans or we talk about other animals, we cannot, our body, our cells cannot make its own food. It needs to get food from some food source. The food that we eat, the fruits that we consume, the supplements that we take, we're deriving our source of food from all these things. We ourselves, our bodies, our cells cannot make their own food. However, plants are those organisms which have the ability to synthesize, to manufacture, to make their own food. So those organisms which can make their own food, they are known as autotrophic organisms. So chloroplast is that organelle which is present in green plants. ठीक है जी? And why do I mean green plants? Because chloroplast has a green pigment called chlorophyll. ठीक है जी? Chlorophyll is a green pigment which this chloroplast consists of. And this chlorophyll is basically the reason why there is this green color to green leaves. ठीक है जी? Now let's draw the organelle, the labeled structure of a chloroplast. So now, second. Okay. So first and foremost, we have our first layer, our first membrane of the chloroplast. Okay. Now I'm drawing another layer, another inner membrane of this chloroplast. Okay, so one thing that we establish at the very start is that we have one outer membrane. This is the outer membrane of chloroplast, and then this is the inner membrane of chloroplast. So one structural identity, one structural characteristic which we have of chloroplast is that it has a double membrane. It has a double membrane. And as we studied in mitochondria yesterday, that the space, the region between two membrane is known as the intermembrane space, which means that the space, the region between two membranes, that space is basically referred to as the intermembrane space. Okay, Jay. Now, let's see what else is present in a chloroplast. So now the chloroplast has its own circular DNA. ठीक है जी. So it also has its own circular DNA. It has its own genetic material present inside it. So here is its circular DNA. Okay. Now, we also see that chloroplast has ribosomes. Now, I would want any one of you to answer this question, that which type of ribosomes would be present in a chloroplast? Would they be 80S ribosomes or would they be 70S ribosomes? Exactly. So in a chloroplast, we have 70S ribosomes present. Now, okay. Another thing that is present in a chloroplast is a structure like this. Okay. And then it also has something on its surface. Now, before I mention what I have drawn, I want any one of you who was present in yesterday's class to tell me that these pink particles that I'm drawing on the surface, we also studied these in yesterday's class in mitochondria. Can any one of you recall the name of this pink structure? You are close, Hajra. They are stalked particles and not stalked membranes. They are called the stalked particles. And if you remember, these stalked particles, I said there is this enzyme, जो इनका head होता है, the head of these stalked particles, they are basically an enzyme. So do you remember the name of that enzyme? Very good. So basically the head, basically this circular region, this pointed region, this is the head of the stalked particle. And this head itself is basically an enzyme called ATP synthase. Okay, now let's take a pause and focus on the structure that I've drawn. So basically, if you could see, it's a stack of circular, or you can say it's a stack of spherical, coin-like, disc-like shape. So I can say if I draw a zoomed version of this, it is something like this. So we have a lot of spherical, disc-like species. So I can say it's basically a pile of these discs or a pile of these spherical species. So what exactly is this spherical species जिसको मैं pile up कर रही हूँ? So this spherical species is basically, if I draw one of them, this one disc, this one spherical species is known as thylakoid. Okay? So basically, there is a stack of these thylakoids, there is a stack of these coin-like, these disc-like species, and this one disc, this one what do you say, this one species जिसका मैं pile बना रही हूँ, it's basically one of them is called thylakoid. And a stack of thylakoids is basically known as something called, the singular is granum, and the way I've drawn two granums over here, so the plural of this pile would be granum. So let me repeat myself. Over here we have a pile of disc-like species. जैसे मैं एक के ऊपर एक बुक्स बहुत सारी रख दूं, उसी तरह से मैंने बहुत सारे क्वाइंस या बहुत सारी डिस्क स्टैक अप कर दिए हैं, पाइल अप कर दिए हैं, और इस पाइल को कलेक्टिवली एक सिंगल पाइल को ग्रेना कहते हैं, एंड more than one or more than two grana are referred to as granum. Okay. So basically, this over here is granum and this over here is granum. So the singular is granum and the plural more than one is called grana. Okay. Now, one single disc, if I draw it like this, is referred to as thylakoid. ठीक है जी. And on this membrane, on the surface of these thylakoids, जो मैंने coin-like structure या जो मैंने disc-like structure draw किया, इन thylakoids की surface पर, इनकी membrane पर, our stalked particles are present. ठीक है जी. So we have our stalked particles present on the surface of each thylakoid. And we know that the head of the stalked particle is basically an enzyme called ATP synthase. Okay, ji. And yesterday, when we were talking about ATP synthase in the structure of mitochondria, we said that ATP synthase is basically the enzyme responsible for the synthesis of ATP. Now, what do you guys think? In chloroplast, is ATP synthesis taking place or not? Is this stalked particle, is this enzyme ATP synthase producing ATP or does it not produce ATP? What do you guys think? Very good, Hajra. So it does produce ATP. Another question arises that chloroplast is basically responsible for photosynthesis. Why? Because it would trap light and it would convert that light energy into into glucose, basically C6H12O6. Because हम लोग बचपन से जो photosynthesis की equation पढ़ते आ रहे हैं, इसमें in that we understand that the raw materials, the carbon dioxide and water combine together to form carbon dioxide and glucose. So where is ATP coming? So basically, photosynthesis occurs in two steps. Photosynthesis occurs in two steps. And what do I mean by that? So basically, photosynthesis is what? It is basically the synthesis or manufacture of carbohydrates due to light. Because photo means light, synthesis means making. So basically making of carbohydrates or manufacture of carbohydrates due to the presence of light or because of light energy is photosynthesis. But is light present 24/7 around us or is light present 24/7 to a plant cell? No. We have a daytime where sunlight is present and then there is a nighttime where the sunlight is absent. So does that mean that a plant or a plant cell is only synthesizing food when light is present? No. The plant does have the ability to manufacture or synthesize food in the absence of light as well. So what happens is that photosynthesis takes place in two steps. One of those steps is the light dependent reaction. And the other is light independent reaction. And this light independent reaction is also sometimes called the dark reaction. So basically what happens is that photosynthesis, for it to complete, it needs these two steps. One step is basically where it requires the light for the process to take place. And in this light dependent reaction, ATP synthesis takes place. Why? Because when this light energy is not basically the light is converted into ATP, and yesterday we understood that ATP is the energy currency of the cell. So that light energy is converted into ATP, and then that ATP is used to make or manufacture carbohydrates. So now tell me, if ATP synthase is present on the thylakoid, or you can say that a stack of thylakoid is known as the granum or the grana. So what do you think? Is the ATP, is light dependent reaction the reaction which is producing ATP occurring on the thylakoid or on the grana, or is it occurring in this remaining region? And by the way, this remaining region is known as the stroma. This basically is the stroma. So tell me, what do you think? The light-dependent reaction would take place on the thylakoid membrane or on the grana, or would it take place in the stroma? Right. So the light-dependent reaction would take place on the grana, which is basically the stack of thylakoid. Why? Because thylakoid membrane has what? It has the ATP synthase enzyme, and that enzyme is basically responsible for ATP synthesis. And light dependent reaction is basically converting light energy into ATP. ठीक है जी. So our grana or our thylakoid is the site of light dependent reaction. So let me write over here. This is basically the site of light dependent reaction. Okay. So RXN is basically the short for reaction. So basically grana or thylakoid is responsible for our light dependent reaction where ATP is being formed. Now, once that ATP is formed in the stroma, which is our remaining region, in the stroma, our light independent reaction would take place. So this is basically our site of light independent reaction. So over here, the ATP would go, and now this ATP would be used to manufacture carbohydrates, our glucose. So basically, chloroplast is present in green plants, ठीक है? अच्छा, हमारे पास जो even the chlorophyll pigment, हमारे पास जो chlorophyll pigment होती है ना, that is also present over here. So our chlorophyll pigment is present over here. Okay, in this region, our thylakoid and our grana region. Also, another thing, if thylakoid or if the grana is the region where the light dependent reaction is taking place, so tell me, हमारे पास जो light absorb हो रही होती है, or the light which our chloroplast is absorbing, would that be absorbed by the thylakoid or would that be absorbed by the stroma? What do you think? हमारे पास जो light energy trap हो रही होती है वो कहाँ पर trap हो रही होती है? In the grana or the thylakoid region or the stroma? Very good. So this is where our light energy is absorbed or trapped. Okay. Is this clear so far? Any questions regarding this? We've understood basically the अच्छा हमारे पास बहुत अक्सर होता है कि हमारे पास जो ग्रैनम होते हैं वो आपस में इंटरकनेक्टेड होते हैं, जैसे यहाँ पर आई हैव ड्रॉन दैट दिस बेसिकली this species is connecting to grana. So this is basically the connecting link, the connecting line between two grana is known as intergranal lamellae. So it's not a very important labeling, but just for your info, I'm writing intergranal lamellae. And as the name suggests, it basically means that it is connecting two grana together. ठीक है जी? So two granum, when they're connected together, the connecting link, the connecting organelle is basically known as intergranal lamellae. Now, chloroplast itself has its own 70S ribosomes, it has its own DNA, so it is also again a self-replicating organelle just like mitochondria. Why? Because it has its own genetic material and it also has 70S ribosomes, so it can have its own, it can undergo protein synthesis and it has its own protein machinery. So it is also a self-replicating organelle. Okay, Ji? Now, let's have a quick look. Chloroplasts have the ability to convert solar energy, or you can say light energy, because sometimes हम लोग अपनी solar energy ना भी अगर present हो, so we can use our light energy and we can cause a plant to undergo photosynthesis. So chloroplasts have the ability to convert light energy into chemical or food energy by the process of photosynthesis. Our granum is the site for light-dependent reaction. And although you do not have to know the exact name of these light-dependent, light-independent reaction, that is your A2 knowledge, but just so that you have an idea, the light-dependent reaction is known as photophosphorylation. And then stroma, which is the site for light-independent reaction, or also known as the dark reaction, is also referred to as the Calvin cycle. So our light-dependent reaction is named photophosphorylation, and light-independent reaction is named Calvin cycle. And granum is the site for light-dependent reaction, and stroma is the site for light-independent reaction. Okay, that's about it for our chloroplast. Any questions about it so far?